Hello everyone. Today I'm going to be going over a little bit of an introduction to the immune system. And this is going to go over the various cellular components of the immune system. And I'll take you through what we call the innate and adaptive immunity in the body. And just to start out with, when we say innate, those are things that are present generally in a normal, typical individual at birth or shortly thereafter. And then we have the specific cells of the immune system, which we refer to as adaptive or acquired immunity. Different cell types. So if you want to check over these learning objectives, first of all, we're going to go over the various components of the innate, the born that way immune system, and then compare the innate to the specific acquired immune system. We'll go through some cell types that are involved in both the acquired and innate immune system. And once we get to those cell types, which we refer to as lymphocytes, cyte meaning cell and lympho meaning of the lymphatic system, we'll go over how they respond differently to specific cells in the body that become infected. There are a couple that we'll go over, the first being T cells, and then B cells, and then what we call the natural killer cells, which are more non-specific. Finally, when we talk about the B cell family, they're associated with the production of signposts to recognize infected cells called antibodies. And those antibodies have a specific structure depending on which infection that is, if it's the first interaction with a pathogen or the second interaction. So just to go over that very quickly, pathos is a Latin root which refers to disease. So a pathogen is anything that is foreign in the body which can cause disease. When we speak about innate or non-specific immunity, again, those are things that are naturally occurring in normal individuals with a normal anatomy that are present generally at birth or times shortly thereafter. And those are going to include some general things. First of all, there's anatomical features and things like the respiratory system or the respiratory tract, which includes ciliated cells which move mucus, which becomes more viscous when there's an infection. And the process of these ciliated cells, and so you might remember that cilia uses ATP to make a whip-like movement in one direction. And cilia's job is to move mucus. So I drew some green mucus here. And that process is called clearance. That is something that happens normally because that's cilia's job to move mucus up from the lungs, up the trachea, and in normal situations it just moves down to the stomach where it's destroyed by the acidic environment. You might know that when you become infected or get sick that your amount of mucus is upregulated and it becomes more viscous, and that might overwhelm the naturally occurring amount of ciliated cells in the body. Therefore, when mucus overrides that, you have to engage in your own clearance, which is referred to as coughing. So just the anatomical features of the respiratory tract, including hairs in the nose and cilia and mucus to trap inhaled particles, will work as a nonspecific barrier to infection. There's also nonspecific enzymes included in various secretions in the body, so tears, prostatic secretions, cervical secretions, respiratory secretions, one of them being lysozyme. And lysozyme is a nonspecific bactericidal, meaning it kills bacteria without impunity, enzyme. That is present also in sweat, and that leads us to the next part. Not only is skin a physical barrier that coats the body and prevents entry of various pathogens, we also have sebaceous secretions in sweat which contain active bactericidal enzymes. Then we finally have the commensal microorganisms, which are more commonly referred to as the good bacteria or the microbiome. And these are present at various entry points to the body, including the vaginal tract, the gastrointestinal tract, and also on the skin. And when there is an imbalance in the microbiome, for example, if somebody is taking antibiotic medication, because they kill bacteria or pathogens without impunity, or yes, bacteria without impunity, it can also lead to destruction of these healthy or good bacteria of the microbiome, leading to a destruction of them, and then what we refer to as a supra infection. So an infection on top of an infection, where we kill the naturally occurring cells and allow an overgrowth of cells like yeast. In another lecture, I go over the inflammatory response, which is something that occurs before the immune response, and we refer to that as inflammatory, but it is a really helpful response, and it prevents the immune system from being activated, because an immune response really takes a lot of energy and can cause some tissue damage. So if the inflammatory process is unsuccessful in removing the pathogen, then the immune system becomes activated. One thing we will go over today is the complement cascade. That's a naturally occurring mediating cellular pathway in the body, which leads to a manufacturing of a membrane attack complex. And we'll go over that in a little bit. So these are things that are naturally occurring that hopefully prevent a pathogen from infecting the body. If those various mechanisms are unsuccessful, then we do have the engagement of the immune system. So before we go over the various parts, let's start this with first reviewing two different cell types that can become infected. And we'll just break them into two very general classes. We first have cells of the immune system, or those can be referred to as specific things like macrophages, dendritic cells, but anything with phage on it, phage means to eat, those cells are engaged in policing of the body, and it's their job to sample the the cellular environment to see if there's anything abnormal there. And if there is something abnormal that can cause infection, those can become infected. And then we have normal cells of the body, or what we refer to as self cells. Any cell in the body can also become infected with a pathogen. And in order to alert the body to the fact that these cells have become infected, they deal with it in two different ways. When an immune cell specifically, so something that is in charge of policing the body, looking for various pathogens, when it encounters a pathogen, it is going to take that pathogen in, do its very best to digest it in the lysosome, so the stomach of the cell, and then it's going to display a portion of that pathogen on something very similar to a flagpole. And we refer to this flagpole as an MHC. We'll go over that more in a little bit. And on that flagpole is a flag. And that flag is going to be a portion of that pathogen that has been broken down. So let's pretend it looks like that. Once we have this processing by the cell, we are going to refer to this now as an antigen, not a pathogen anymore. A pathogen is the disease-causing organism. Once it's been processed and displayed, we refer to it as an antigen. That now means the cell has a special name called an antigen-presenting cell. When we're looking at these specific types of trafficking cells or policing cells, the flagpole that they display to the immune system is called an MHC type 2 complex. When a normal cell becomes infected, we still have a similar process whereby the pathogen is brought inside of the cell, digested in the lysosome, and then processed in a very similar manner. However, what we see in this pathway, when it puts up its flagpole, it's a different color. It looks different. And that is called an MHC class 2. And that alerts the immune system to the fact that now we have a normal body cell that's become infected. However, we still are going to have the display of this portion of the antigen to the body. It just is going to use a different flag or a different flagpole to do that display. These are going to engage the immune system in two different ways, and let's start going over that now. When we talk about specific immunity, I mentioned that there are three different types of cells. We've got our T cells, our B cells, and our natural killer cells. All of these cells start from stem cells, which exist in the bone marrow, and they are processed into the three different types of lymphocytes through a maturation process. These cells flow throughout the lymphatic system, which is a channel pathway usually associated kind of like a sewer system picking up the garbage. And the lymphatic system contains various organs like the spleen and Peyer's patches, the thymus, and various lymph nodes, which are present throughout the body. And as T cells, B cells, natural killer cells flow throughout the lymphatic system, they are sampling this area, looking for pathogens that can then be destroyed or encountered by these various types of cells. And when these cells, T, B, and natural killer cells, are flowing throughout the lymphatic system and engaging in an immune response, they have four jobs to ensure that everything works appropriately. The first is being able to recognize a foreign body or recognize a cell that has become infected and is displaying its flag and flagpole, learning which cells match that specific antigen so that it can continue to fight it, and then also keeping the memory of what that antigen particle looks like, at least the surface part of that cell, which we refer to as an epitope. So remembering that generally for years to decades at a time. And the fourth thing that it requires is self-discrimination, meaning that it is able to recognize physically the difference between a cell that's become infected versus a normal cell in the body. We only want cells of the lymphatic system, these lymphocytes, attacking cells that have become infected. If they start attacking cells that are not infected, normal body cells, we refer to that as an autoimmune disease. Auto, a prefix meaning self. In that case, we have the destruction of normal, healthy body cells, which leads to disease. So it's very important that these lymphocytes know how to attack only cells that have become infected. So let's start going over these different types of cells. We'll start out with T cells, and they are named T cells because not only everything is developed in the bone marrow, but they mature in the thymus. Thymus starts with T, so they were named T cells. B cells are produced in the bone marrow, and they mature generally in the bone marrow in humans. However, when they were first discovered, they were discovered in a part of the body of a seagull referred to as a bursa. B for bursa is why they were named B cells. Humans don't have bursa, but we still like to respect our seagull neighbors by maintaining that name. Finally, we have the natural killer cells, which definitely have the best name, and they're more nonspecific. They don't necessarily respond to an MHC complex like T and B cells, but they do have their own jobs with tumor surveillance and being able to spot cells that have become infected with a virus. So starting out with T cells, when we speak about T cells, we refer to this as cell-mediated immunity, meaning that when a T cell system is activated, it's going to use other cells to destroy the cell that has become infected. A T cell in real life, this is a real T cell, and it looks a little furry in this picture. And what you are looking at are things, very easy name, they're called T cell receptors. And T cell receptors are going to hopefully recognize a surface characteristic of that antigen that's been displayed to the body. And that shape, so we might look at this portion, pretend this is an antigen fragment, that shape is referred to as an epitope region. And you might remember, or if you don't know, now we're going to learn that things in the body are attracted to one another by their shape. The more closely something matches another thing's shape, the more likely they are to have a binding interaction. That is called affinity. Affinity is speaking about attraction. So when a cell displays an antigen fragment, the hope is that throughout T cells moving throughout the circulation, at one point a T cell receptor will have a shape that is close or matches the epitope portion of an antigen fragment, and those two things will bind. And that's what you see here. We have an antigen-presenting cell. Remember, an APC is a cell that is usually a phage cell, like a macrophage and a dendritic cell, whose job it is to police the body for foreign material. It processes that, makes its flagpole, its MHC class 2, and then presents the flag, which is an antigen fragment. There's billions and billions of T cells that flow throughout the lymphatic channels, and then also will flow through the bloodstream as well. And they all have their own unique receptors. When a T cell floats by that matches very closely in shape the epitope portion, those two things will bind. And that's where you see the beginning of the immune response, a T cell having a binding interaction with an antigen-presenting cell. And if we look at a molecular level of what these T cell receptors look like, they're embedded in the plasma membrane. There is a constant region in the center, and what varies between cell types and the variable region is the shape of the antigen binding site. So the closer things are in shape together, the more likely they are to bind. So that is the T cell's job, to express a T cell receptor and bind to an epitope portion of a displayed antigen fragment on an antigen-presenting cell. There are two types of T cells, and they also have pretty straightforward names. The first cell is referred to as a T helper cell, and we refer to those as helper cells because they help start the immune response. They are the things that have a T cell receptor on them that match, hopefully, an epitope region of a displayed antigen fragment. When those bind to the matching region on an APC, that engages the rest of the immune response. So T helper cells are the ones that help start the immune response. We refer to these as also CD4 positive cells because CD4 plus is the surface protein that coats T helper cells. So that's just what covers them. And then there's two different types of T helper cells, which we'll see in a little bit. The second type of T cell is called a cytotoxic T cell. Cyto is cell and toxic means deadly too. Cytotoxic T cells are also called killer T cells, which is a much better name, killer T cells. And killer T cells are engaged in what's called cell-mediated cytotoxicity. They bind to cells that have become infected and they spray kind of like a very poisonous substance on them so that it dissolves and kills that cell. There are two different types of killer T cell classes, 1 and 2, which we'll see also in a little bit. But the main thing that you should remember just generally about them are that the TH or CD4 positive cells, those display MHC class 2 because they're generally the policing cells of the body, whereas cytotoxic T cells, also called CD8 positive cells because they have a different surface protein expressed on them, they display MHC class 1 flagpoles. That means that CD8 positive cells are responding to cells that are normal in the body that have become infected, whereas T helper cells are responding to things like macrophages who are on the lookout for foreign material. So they're called in to start this process when there's foreign material, whereas killer T cells are engaged in this process when normal cells of the body become infected. Just going over this one more time, T helper cells. Let's look here. Here's a CD4 positive cell. It is expressing an MHC2 complex and responding to this MHC class II complex expressed by the antigen presenting cell. Here's our T cell receptor, and this portion of the T cell is going to bind to the antigen presenting cell and then engage in the rest of the immune response. Whereas in cytotoxic T cells, they respond to MHC class I proteins. We still have our T cell receptor, but they are responding to normal cells in the body, and they help destroy cells that have become infected. When we take a closer look at this binding interaction, so this says any cell, which is referring to a normal cell of the body that's become infected, we can see here it's MHC class I protein. We see our antigen fragment, and then we see the T cell receptor with the closest shape to that fragment, which now has a binding interaction. When we said the surface protein, here's a CD8 surface protein, that also is going to assist in that binding interaction. So we have the antigen presenting cell binding to the T cell receptor facilitated by the surface protein. The other arm of the immune system is going to involve cells referred to as the B cells or B lymphocytes. B cells look a little bit different. They obviously don't have T cell receptors because they aren't T cells. Instead, they are covered with antibody. Not all antibody are Y-shaped, but in this picture they are. And here is a B cell covered with antibody that in the very same way we have a T cell receptor binding with an epitope region. The antibody complex ends in a shaped type of structure, which also recognizes that epitope region. We refer to B cells as part of the humoral immune system rather than the cell-mediated, instead of using other cells to help fight an infection. It secretes antibody in a liquid form, and humors or humoral types of things in the body refer to liquid. So when antibodies are secreted, they're in a liquid form. When we have B cells being activated, they diverge into two different things. One of them are memory cells, which will remain coated in the antibody with the same shape of the epitope region of a pathogen. And those can live for decades at a time. And that really forms the basis of lasting immunity and sets the framework for vaccination. Plasma cells are the other diverging cell type created when B cells are stimulated, and plasma cells are the active antibody factories in the body. So B cells themselves, it's their job to bind to an infected cell by this portion of their antibody, but then they turn into memory cells and plasma cells. And plasma cells are the ones secreting that antibody, 2,000 antibodies a second. And remember, in the lymph nodes, there's high concentrations of these various immune cells. And because antibodies are liquid, we do see things like swelling of the lymph nodes during infection. And a lot of that has to do with this really large production of liquid antibody. Finally, we have the natural killer cells, which are more specific. It's not their job to bind to a specific epitope region. Instead, they have 3 different functions that they engage in. Here are some real-life natural killer cells attacking a Neisseria gonorrhea molecule. So you can see that they've been activated, meaning that they change their cell structure and grow little cell feet. And they can move to attack and engulf cells that have become infected. There's three ways that they function. The first is called antibody-dependent cellular cytotoxicity. The rule that I like to think of with antibodies, an antibody is a nice way to tag a cell to tell the body that it's been infected. So whenever a cell has an antibody on it, that tells the various cells in the body that that cell's infected. It's very similar to putting a Post-it note onto something to saying that this thing is garbage. So let's say we have a chair in the classroom and you want it thrown out. At the end of the day, when housekeeping comes in, if you just have this chair in a line of other chairs, how will they know that that's garbage? Well, there's no way to know unless we put a note on it that says garbage, right? This thing is broken. So if you put a little note on it, that alerts housekeeping to say, all right, this chair is broken. There's something wrong with it. I will also trash it. And that's the way antibody work. When antibody is on a cell, we know, the cells in the body know, that it's infected and it's okay to kill it. When the cells move in to kill that cell, that's called antibody-dependent cellular cytotoxicity. So killing cells when it's covered in antibody. Very proficient system. It works as long as self-discrimination is evident, meaning that this cell, the natural killer cell, knows the difference between a pathogen, like this Neisseria gonorrhea, and a normal cell like a red blood cell. It's also a job of natural killer cells to look for various hallmarks in cells that they engage in when they become abnormal, like in the tumor process. This works at a very small level and with certain cancer types. And if natural killer cells did their job and work very, very efficiently, there would never be any cancers in the body. There is a large arm of individuals who believe that cancers in general are a product of a malfunctioning immune system. And that is a very well-supported theory now and lays the framework for the immune research that is going on at many cancer institutes. The final way the natural killer cells work is detecting cells that have become infected by a virus. And this is the trickiest thing because when a cell becomes infected by a virus, the virus is going to go in and colonize that cell and use the parts of that cell to just make more copies of itself. So it turns that cell into a virus-making factory. But why that is tricky is you cannot tell from looking at that cell from the outside that it has virus in it. There is only one way where we can really distinguish between cells that have a virus inside of them and one that do not. And that is by their ability to display an MHC complex. So a normal cell of the body, remember, can display an MHC complex, and it tells the other cells in the body that it's become infected. So that is going to cause T cells to come to the area. When natural killer cells are called to the area, they're like the snipers of the body. It's their job to kill anything that they're there to kill. It's not their job, remember, to bind to MHC complexes and epitope regions. Instead, their job is to kill things. When they're called to the region, they know that there's things infected there. It's kind of like the cops being called to a gunfight. They know that there's bad stuff happening there, so they're going to respond in a certain way. Natural killer cells are policing this area, and when they find a cell that doesn't display an MHC1 complex in a sea of cells that have become infected, it recognizes that as a virally infected cell, because that is really the only way we can tell that there's a virus inside. If there's a virus inside of the cell, it cannot display an MHC1 complex because its cellular organelles that would typically break that down and display on a protein fragment the antigen have now been taken over to make the virus. So natural killer cells are really the ones that engage in looking for and killing virally infected cells. To finish, we'll just go over antibodies. We've mentioned them before. Generally, when antibodies are drawn in a picture, they are Y-shaped. And there are some that are Y-shaped. There are some that are dimers, and there's some that are pentamers. However, we will see that the regions of antibodies remain static, regardless of which type they are. There's two various regions that we should pay attention to. The first are the antigen binding sites, referred to as the Fab portion or the antigen binding fragment. And then at the feet of the antibody is the Fc portion, the crystallizable fragment. At the antigen binding sites, remember, those are just like T cell receptors going to bind to epitope portions of a displayed antigen. So here we have antigens of varying shapes and the corresponding antigen binding sites. Remember, the closer they are in shape, so here this yellow antigen is very close in shape to this portion of the antibody, those things can bind. There are three different subtypes of antibodies. We have a pentamer, so that's 5 sides. We have a dimer, which is 2. When things dimerize, they make a mirror image of one another. And then we have just a single antibody, a Y-shaped monomer. IgG, IgD, and IgE, which stands for immunoglobulin type G, D, or E, are all monomer shaped. IgA, immunoglobulin A, is a dimer, and IgM, which is the first antibody produced by plasma cells when there is a contact with an antigen, is the only pentamer that we have. I like to think of it as a ninja throwing star because it's the first thing that is produced by these cells when they really don't know what else to do. They just produce a large amount of this immunoglobulin M, hoping that these antibodies can bind to and then wipe out this infection. Here we see the subtypes again. You can see where they are produced and when. Again, IgM, immunoglobulin M, first things produced by activation of B cells, by plasma cells when there's infection. That's our pentamer. IgG, that's the principal immunoglobulin in blood. This crosses the placental barrier during pregnancy and can give the fetus a little bit of lasting immunity for about 6 months after birth. There's IgE. IgE is what you receive a test for in allergy tests. So if you're allergic to peanuts, for example, that would be confirmed by finding an IgE against fragments of peanut. We have IgA, that's our only dimer. And IgA is found in various secretions like breast milk, saliva, respiratory secretions, and GI tract secretions. So that can help limit things from coming in at these various entry points. And then finally, we have IgD, which is a monomer, and that's an antigen receptor, which really just coats lymphocyte membranes. The other thing about IgE, which I mentioned very briefly, is that it is mainly associated with allergies now. It is something that evolutionary biologists believe that developed to help fight and recognize parasitic worms, but in places where there is good drinking water and good sanitation systems, we see low levels of parasitic worm infections, meaning IgE really does a lot of binding of harmless substances like pollen or cat dander and engages the immune response to make you miserable and have these hypersensitivities. We'll talk about it in another lecture. So antibodies, again, they can do a couple of things. They can sit on cells like macrophages and mast cells, which are responsible for releasing histamine in the body, and that helps facilitate this process. So here's a pathogen, here's a mast cell with an antibody on it. So these antibody can bind to expressed epitope regions on the pathogen, which helps enhance phagocytosis. We refer to this as a loading or priming of mast cells. That makes them hyper-responsive and also means that the inflammatory response lasts a shorter amount of time. Also, we can see antibody just binding to these epitope regions. And remember, if there's an antibody on something, then it's tagged for destruction. That is going to allow cells in that area, like cytotoxic T cells, to come over, engulf it, and kill it. The other thing that antibodies help with, you see here in the center, it says complement binding region. Antibodies help trigger the complement cascade. We use this term cascade when something starts, kind of like a waterfall. Once the water goes over the edge, that's a great looking waterfall, we can't stop it. It's just going to keep going. And that's why cascades work. Once one step happens, then it's going to go through to the end. The complement cascade, in biochemistry, you have to know every single step of it. But generally, the complement cascade is activated when this antibody portion becomes active, and there's a ton of steps. There's like 20 steps in this complement cascade. What is important to know is that at the end of the complement cascade, we have the creation of something called the MAC, the membrane attack complex. When the MAC is created, it can either be inserted into a normal cell of the body that's become infected, and it just forms a hole. And when cells have a big hole in it, they can't function. Same way with you. If you had a big hole caused by a shotgun wound or a shark bite, your body isn't going to function normally either. When cells have a big hole in them, they lyse, so they break apart and they die. So this membrane attack complex is assembled by the various steps of the complement cascade. The other thing that it's helpful for is called opsonization. And opsonization is really the creation of this membrane attack complex, but specifically in bacterial cells. So if a bacteria, we'll make it like angry looking, gets into the body, this can be recognized by an antibody, right? The antibody can sit on it and alert the body that it's infected. And it can also engage in the complement cascade. So it activates it and then inserts a membrane attack complex into the bacteria, and that allows things to enter, and then the bacteria dies. To leave things for the next lecture, which will go over the various steps of the immune response in detail, when we think about antibody, we want antibody to be as efficient as possible. We want it to match the epitope region of the pathogen, so match the epitope region, and we want it to do that quickly. The other thing that we look for with antibodies is an antibody test. So you may have heard of this. This is the COVID time, COVID-19, and if you hear the scientists talking on the news, they will refer to different tests. They'll say there's a test to see if you were exposed and a test to see if you've already had it and now you're possibly immune to it. When, again, the body first encounters a pathogen and it doesn't really know what else to do, it makes its little ninja throwing star, its immunoglobulin M, throws it out and just kind of hopes that things go well. That's the initial response. Once the body learns what that epitope portion looks like, it can make a specific copy of that epitope portion. So here we see this structure. Here's our IgM. And once the body learns what this pathogen region looks like, it can choose what portion it specifically needs and then makes copies of that. So once there is the production of IgG with a matching antibody receptor to an epitope region, then we know that somebody had a previous infection and may hold lasting immunity to that substance. When there is a secondary exposure to a pathogen, we see the production of really large amounts of IgG. There's a small amount of IgM to begin with, and then a switching to IgG. We refer to this as class switching. That means that the body has learned to recognize that specific antigen. It goes from making IgM to making IgG very quickly. Whereas the first time, again, that the body comes into contact with a pathogen, we just see a large amount of IgM that's created because that's what the body knows how to do. All right, I hope you learned some things about the various parts of the immune system, and in the next talk, we'll put them all together. If you want to take a look at these questions, we'll go over them very quickly. In a primary immune response, activated B cells and then their pathways, including plasma cells, the antibody factories, produce mostly IgA, IgD, IgG, IgE, or IgM. We have our pentamer, our ninja throwing star. That's our primary infection. Which of the following can secrete antibody? Is it B cells, helper T cells, killer T cells, natural killer cells, or plasma cells? If you say B cells, you'll make your microbiology teacher very sad. T cells do not make antibody. We can get rid of them. Natural killer cells, they definitely don't make antibody. Plasma cells, those are the antibody making factory of the body. Come back for more videos. We will talk more about the immune system, and feel free to leave any comments or questions you might have.